good about your learning. It makes you feel really good, but actually all you're doing is recognizing all the words which you've been able to do since you were 3 or 4, and then remembering those words as you read them, rather than actually storing them in your long-term memory and linking them all together. So my preferred way of studying is have a really good textbook, or watch a good quality video, and make my own notes as I go through it, and then have a little go at maybe rewriting those notes from memory, see which bits are missing, see which bits I have managed to store, and go through it that way. So we're going to do plant tissues, and there's quite a lot of tough key terminology. So if I just list for you a list of tissues that it's worth you knowing a little bit about, we've got the epidermal tissue. We've got your palisade mesophyll. We've got your spongy mesophyll. We've got the xylem and the phloem. And then we've got your meristem tissue. So depending on where we get to today, it is worthwhile you making sure you've got some notes on all of those bits and what they do, then how they connect together and how those work as systems. So these are the tissues. Remember, different types of tissues grouped together to form organs, and then a group of organs will then be grouped together to make an organ system. Okay, so I guess the first thing we could do is we could very quickly sketch a section through a leaf, and this is a cross section. So imagine we've sliced through the leaf and we're now looking inside it, and it is magnified. So we've sliced through the leaf and we're looking inside. So I'm just going to do a very quick sketch. It's not going to be particularly neat. You can definitely go away and find some really lovely, certainly artistically more accurate diagrams than I'm going to do. But I'm just going to do a quick sketch and try and get as many labels on here as I can, so that we've got a good idea of what's going on in our leaf. So we've sliced through it. This is the top of the leaf. So if I'm looking down, this would be the top. And then we've got this little layer of cells here. Okay, and I'm going to take a liberty. I'm just going to put one nucleus in there, but they're probably all going to have a nucleus. And then we've got this sort of beautiful, slightly different shape stack of cells. And then just break them up a bit. And then again, this lovely stack of cells here. They look like bricks, sort of stacked together. And I'm going to try and make these stand out a bit by showing that they've got a high number of a certain type of organelle inside them. There we go. And then we're going to have sort of a little bundle of cells here. All that quickly, and then some sort of larger, more irregular shaped cells here. And then again, a nice final layer at the bottom here. So if you have a little go at that sketch for me, can you then do that sketch very quickly now? And can you label as many of those tissues that we started with on here? So we've got these tissues. Where are they all found on here? There's only one that you won't be able to put on, and that is your meristem, and that's because the meristem is only found in the tips of the shoots and in the roots. So it's in the parts of the plant that are growing, and they're growing quickly, and we're able to see lovely mitotic division in those areas. and that's where the meristem is found. So that's the only tissue you're not going to find here. So let's have a look. Go. Can you try and label as many of those bits of tissue? I'm going to start showing you where I'm going to put some labels. So I'm going to put one here. I'm going to put one here. I'm going to put one here, and I'm going to go there. Okay, so I've definitely got those 4 labels. I think I probably know what that is. I've tried my best over here. Just going to draw the label like this to draw something slightly different. And can you tell me if you can identify that organelle that I was desperately trying to draw? Lots of them as we went along there. So can you have a little think about those labels that we had, those types of tissue at the top there, and where they would be found on my diagram here? I'll give you some first letters. I'll try and finish this bit. I rushed that a bit. I need to just put some more cells in here. That's the best I've ever done there. Great. Okay, 1 more minute. See if we can get all of those on there. So we remember we said this is the top of the leaf. So this is where the majority of the light is going to hit, and therefore this is the upper epidermis. And then by elimination, that could just be the lower epidermis. This is your palisade mesophyll. And you've got this lovely regular shape, like a stack of bricks all next to each other. And underneath that, you've then got your spongy mesophyll. And this has got lots of air spaces. So although, yes, my diagram is unfinished, stick another cell in there. I have purposefully left these spaces because we need to be able to move air in and out in order to, when we need a gas, it's able to diffuse in, and when we create a higher concentration of gas, it's able to diffuse out. And that process is happening through this space here. I don't know, was I pointing to the cell? Yes, I was definitely. So that arrow went to the cell rather than to the gap itself. So this is a guard cell, special type of cell that is going to be able to open and close this space here by expanding and contracting. Remember, this is your stomata. So the actual gap is the stomata, and the cells that create the gap and the cells that make the gap bigger and smaller are called your guard cells. This bit here, this was my not particularly good attempt at a vascular bundle. And within that was my xylem and my phloem. So if you just went for those 2 labels, perfect, that's well done. But the whole thing together, and you'll often find, you'll find the xylem in one shape and the phloem in a slightly different shape. They'll be in this sort of bundle running through through the leaf. And then my final little label up here was of my organelle. Mustn't forget that, my chloroplast. So it's worth being able to identify where all of those parts are, because actually we need to know a little bit about what each of those is for, what its function is, and how it's adapted to that function. So if we have a little think about the adaptations of a leaf, how it is adapted to do all the processes efficiently, we could have a little think about some adaptations. And whenever we're talking about anything biochemical, if we say surface area, then we're going to be correct about something along the way. So they've got a large surface area, and that increases the surface area for diffusion of CO2. And also just increases the surface area for the absorption of light. The leaf is also very thin, and that means that CO2 can diffuse quickly to the palisade mesophyll where it is required. So if we think CO2 out here diffusing into our spongy mesophyll, well, actually it hasn't got very far to travel because the leaf itself is a very thin structure. So it's thin so that CO2 can diffuse to the palisade mesophyll. Excuse my terrible abbreviation. That's definitely not allowed in your exam. Don't do that. But just for ease, I'm going to write it here. We've also got these wonderful organelles in high concentration. In large number, we have our chlorophyll inside the chloroplasts. And the chlorophyll is the pigment that is able to absorb light. Remember, this pigment can absorb light in high concentration in our chloroplasts, and the chloroplasts are in high number. We've also got a network of veins running through. And they're able to transport water. Also able to transport carbohydrates. And if we think about the direction of these, the water needs to go to the leaf, and the carbohydrate is being made in the leaf by the process of photosynthesis, and it's going to need to travel around the rest of the plant. So network of veins allows a supply of water to go to the leaf structures, and then for carbohydrate, which is a product of our photosynthesis, to then be transported around the rest of the plant where it is required. We've got those stomata. Remember, the stomata is the actual space created by the guard cells, and this allows CO2 to diffuse in and for the high concentration of oxygen that's created to diffuse out. That's very fortunate for us. That then keeps the O2 levels at the level we require, or we're adapted to live at that level. The epidermis are thin and transparent. So your epidermal layers are thin and transparent, and that allows more light in. On the top layer, we also have a cuticle called a waxy cuticle. And this is able to protect the leaf itself without blocking out any sunlight. So it protects, but also allows sunlight through. We've got our palisade mesophyll layer. Write the whole word this time. Got our palisade mesophyll. And this maximizes the way it's designed, the way these cells are shaped and stacked together, are absolutely optimizing the amount of light absorbed so that they can then maximize the amount of photosynthesis happening. So we've got palisade mesophyll is there to maximize light being absorbed. We've then got our air spaces. In the spongy mesophyll layer, and these are allowing CO2 to diffuse. And then finally, we've got that vascular bundle. Squeeze it in here. Vascular bundle. And this has got thick cell walls that help actually support the stem and the leaf. Some thick cell walls that support the stem and the leaf. Okay, if we just pop back to our diagram for a second. So we've got our adaptations of our leaf. We've actually got the structures of the leaf, and we've been able to label our tissues. Now, if we have a little think about, we've talked a lot about how these adaptations allow for CO2 diffusion. So I just thought we'd clarify that. So we're going to have CO2 in high concentration outside the leaf. So it's in high concentration in the atmosphere. It's going to diffuse. So we're going from a high concentration to a lower concentration. It's going to diffuse into the air spaces. So remember, diffusion is a net movement from a high concentration to a low concentration. These molecules are actually going randomly in lots of directions, but because there are more of them here, then they go to the area where there are fewer of them eventually. And so they're going to diffuse into our air spaces in the spongy mesophyll. And then after that, they're able to diffuse through the cell wall and through the cell membrane into the mesophyll cells. So actually going to diffuse into the cells, mesophyll cells itself. It's going to dissolve in the cytoplasm and then diffuse up to our chloroplast doing photosynthesis. So diffusing high concentration to low concentration in the air, it can then diffuse through the cell wall and through the cell membrane into my cytoplasm and then travel and diffuse all the way to my chloroplasts. Okay, let's talk a little bit about the organs of the plant. So my organs of my plant are my roots, my stem, and my leaves. And they form if we group these together, they become an organ system. And that is the organ system for transportation. So if you think about your cardiovascular system is a group of different organs that come together to transport your blood around your body. In the same way, that if I group together my roots and my stems and my leaves, then I get the organ system that is designed for transport. We need to talk in quite a lot of detail about that transport, and we need to know in quite a lot of detail about the xylem and the phloem. So let's do that next. Let's say functions of xylem and phloem. So these are our two transport vessels. This is the name. These are our transport vessels, analogous to your arteries and your veins and your capillaries. However, these two have really very distinct, very separate roles, whereas your arteries and your capillaries and your veins change in shape around your body, depending on what their role is. These actually carry different substances around the plant and for different reasons. So let's first state what those are. You've got your xylem vessels. And you've got your phloem vessels. So take a second and just see if you can list what sort of things you think are going to be transported by these two. I'll give you a second to think about it. You might be able to remember the most basic definition if you go back to Key Stage 3. You say this one transports one thing, this transports another thing, but actually we can give a little list of things. Okay, 30 seconds. So xylem vessels very specifically transport water and minerals, and very specifically from the roots to the stem and leaves. And the phloem then. Phloem is transporting food materials. And these are mostly sucrose and amino acids. And these are made by the plant during photosynthesis. And we need to transport them to the non-photosynthesizing regions. So they need to travel all over the plant. So this is very directional from the roots to the stem and the leaves. The phloem vessels, these are bidirectional. They're traveling all over the plant in order to take these things, these products of photosynthesis, to non-photosynthesizing areas of the plant. And when you group them together, you group them as a vascular bundle. So if we just go back to our diagram, I did have a go at separating the two. So if I highlight those two now, if we had a section through the plant like this, and we looked at our vascular bundle, all of this tissue up here would be your xylem tissue. And again, it would try and make it obvious. I've not been particularly clear there, but the tissue below them would be your phloem. So xylem would be at the top and phloem would be underneath. Now, we also need to be able to spot how these two look, where we would find these in a cross-section of a stem and a cross-section of our root. So let's do the stem first. So I've just sliced through the stem of the plant. I'm looking down through the top of it and I can see my vascular bundles. Here they are. And check what colours I went for. And in those vascular bundles on the outside, you're going to find phloem, and on the inside of the vascular bundle, you're going to find the xylem. And then we could do the same through the root. Again, I'm going to slice through a root and look down inside it. And in this instance, the vascular bundle is going to be found right in the center. And we've got our xylem tissue right in the center. And we've got our phloem tissue that's around the outside here. So phloem here and xylem here. Okay. The next part of the plant is worth knowing a little bit about are root hair cells. And these are adapted for the efficient uptake of water adapted for the efficient uptake of water, and this is by the process of osmosis, special case of diffusion involving water, and also they're designed to take up minerals or mineral ions, and they move those against the concentration gradient. So they're going from low concentration in the soil to high concentration in the plant. And so remember, to do that, to go against concentration gradient, we have to use active transport. So how are they adapted? Well, they are the root hair cells themselves are single-celled extensions of the epidermal cells in the root. And those projections increase surface area. And that therefore increases the surface area for water to move in by osmosis, and also massively increases the surface area for mineral ions to be taken up by active transport. They grow between soil particles. They're actually able to sort of nestle their way in. And water, remember, is moving in by osmosis, as it is in a higher concentration in the soil. Now, that water movement in then is part of quite an important process in the plant, that we need to know quite a bit about. So we've got the root hair cells, they're doing osmosis, and they're doing active transport. And they are the first part of the process of transpiration. Transpiration is the process where water travels up the xylem from the roots to the leaves and is then replaced by more water behind it. And that replacing of water, that constant flow of water up the plant, is called transpiration. And we can give it a little definition. It's defined as the loss of water vapor from the leaves by evaporation so the loss of water vapor from the leaves by evaporation and what is happening is the water is moving from the mesophyll cells and then out through the stomata. So from the mesophyll cells and then out through the stomata. So here's our little definition: loss of water vapor from the leaves by evaporation. This evaporation is occurring from the mesophyll cells out into those air spaces and then as water vapor out through the stomata. So we need to know a little bit more about the xylem structures and how they are adapted to support this process. So xylem, and these are our adaptations. The cell walls of our xylem cells are lignified. And what this means is lignin is deposited in the cell walls. And it actually causes the cells to die. The cells, these lignified cells, become hollow. So loss of all of their organelles, so no more organelles inside, no more cytoplasm, and they join end to end and form a continuous tube through the plant. And it's this tube that our mineral ions and our water travel through. And the lignin is the thing that strengthens the plant and helps it withstand the water pressure. Okay, so if we've got transpiration and we know it's happening through these tubes, can we have a little think about 4 factors that are going to affect the rate at which transpiration takes place. So what 4 factors are going to have an impact on the rate of transpiration? So if we do a quick table, you could have a little go at filling this in. So I'm going to have factor there on the left, and then I'm going to say what condition that factor might be in. Maybe if it's very high, what happens, and then we'll be able to say the converse. If it's low, then this will happen. And then what is the effect? So let's draw up a quick table. Can you have a little think? Can you think of 1, 2, 3, or are you able to get all 4 of them? So we know that we're talking about water vapor. We know we're talking about evaporation. So let me do the first one for you, and then you have a little go at the next 3. So my first factor is going to be air movement. And I'm going to talk about if air movement is high, if there is lots and lots of more of air movement, what is going to happen to the rate of photosynthesis? Well, if air movement is high, not photosynthesis, excuse me, transpiration, then there is going to be more transpiration. The reason for that is we are thinking about diffusion. We're thinking about diffusion, we're thinking about concentrations, and if we're thinking about concentrations, we're thinking about concentration gradients. And remember, that means the difference in concentrations in 2 areas. And if we maintain a concentration gradient, it means we're maintaining a higher concentration in one area and a lower concentration in another area, and therefore more of that thing is able to move in. Similarly, in your lungs and particularly your alveoli have a really rich blood supply, and that keeps the red blood cells moving all the time. So that means oxygen is able to diffuse it and bind to the red blood cells. And as soon as it's attached to those red blood cells are moved away very quickly. And so new red blood cells turn up without oxygen bound, and that is maintaining the concentration gradient. Well, it's the same here. If air movement's very high and you've got water vapor leaving the plant, that water vapor is moved away very quickly, and therefore the water concentration in the air is lower, and therefore I'm going to get greater water loss. Okay, another second just to try and do the next 3. You have a little go at those. And then we need to talk about an experiment. We need to just very quickly talk through an experiment we can do that will help us test our theory here in terms of these 4 different factors. So I'll give you the first letter of the next one I'm going to do. Obviously, don't have to have it in the same order as me. And I'm actually going to put all of these in a high state. Worthwhile you being able to just reverse this table. Maybe when you go back to do your revision notes, you'll say, okay, 4 factors. I'm going to rewrite them out from memory, but I'm actually going to write in the low state. I'm going to say, well, if it's low, then there is no concentration gradient. There is therefore not going to be much transpiration, because once the water vapor leaves the plant, it stays there and you're not able to maintain a concentration gradient. So maybe when you rewrite this, try the converse. So my next one is humidity. And then I think here I was thinking light intensity. And then finally, temperature. So the 4 factors that I can vary to vary the rate of photosynthesis are air movement, humidity, light intensity, and temperature. So if I keep the humidity high, then my rate of photosynthesis is going to be less. Again, exactly as we said before, there's going to be a weaker concentration gradient. If there is, remember what a definition of humidity is, it's the amount of water vapor already in the air. So if there's already lots and lots of water vapor in the air, then transpiration is going to be slower. There is going to be less transpiration overall because we're not able to then move against concentration gradient. If there's lots and lots of high concentration of water vapor in the air, we're not then going to be able to move out of the plant, so the water is not going to be drawn up through the plant. Light intensity then. If light intensity is high, then I'm going to get more transpiration. And what happens is your guard cells actually respond to light intensity. And when it's high, they become turgid. And the stomata open, allowing more water to be lost. If we think about that, that makes sense because this is a byproduct of the processes happening in the plant, and therefore we want to get rid of any waste and draw more up through the plant. And so when light intensity is high, the guard cells become turgid. They open the stomata, they make that space, the stomata space larger, and allow more water to be lost. And finally, temperature. If the temperature is high, well, going to have more. We think back to our biochemistry, this is to do with kinetic energy, isn't it? The temperature is higher, the particles have more kinetic energy. There's more kinetic energy, therefore things are moving around faster rate at which the molecules are going to diffuse. Okay, very quickly then, we have a little think about an experiment we could do to test this, to see if we change the air movement, i.e. blow a fan on my plant, or increase the humidity, have lots and lots of water vapor around the plant, or increase the light intensity, or increase the temperature, or indeed the converse, then we would need to show that the rate of transpiration changes. And so there's a little practical we can do. And this is going to be a very rough sketch, and I highly recommend you go away and research this practical a little bit yourself. But we can actually investigate transpiration. Investigating transpiration using a special piece of equipment called a potometer. It sounds fancy, and certainly when you look at the setup, it looks a bit fancy, but it is a relatively simple idea. So firstly, we need our plant. Here we go. I'm just going to extend me. I've got some leaves on the plant there. And this stem is going to go inside a tube, and we're going to seal this bit here. So it passes through, but it's sealed. And we're going to go into a reservoir. And this reservoir is all filled with water. And it connects to a capillary tube at the end here. And the capillary tube is filled with water. But somewhere along the capillary tube, there is a bubble. We introduce that bubble ourselves. And then we have a scale here. and the tube, capillary tube, is down into a beaker of water. Okay, as I say, very, very simplistic. There's lots of other stuff that's going on here, but nevertheless, the idea is I've got this bubble here. I've introduced this bubble. All of this solid tube of water, all the way down to the beaker here. And then what I can do is I can increase light intensity. So here's my lamp. And I'm going to increase the light intensity. And I've said in my table up here, when I increase light intensity, the guard cells are responsive to that. They become turgid, they open up, and therefore the plant's going to photosynthesize, but it's also going to lose water vapor. And as that water vapor is lost from the plant, that means the water vapor is drawn up from the beaker along this tube here, and we see the bubble move along the scale. And obviously, if it moves into the water, we then can't measure, into the reservoir, we can't measure anymore, but we see that bubble move along. And we can therefore measure the rate at which the transpiration is occurring by measuring the speed that this bubble moves. And so we can test our 4 different variables, can't we? We could actually investigate each one of these. For airflow, for air movement, we could use a fan, and we could set the fan up blowing across the plant here and see the bubble move. For humidity, we could actually wrap the experiment itself in a plastic bag. And so any water vapor escaping would build up and therefore increase the humidity around the plant, and presumably we would see the movement of the bubble slow. For light intensity, we can move the light source, and it's worth going away and looking up the inverse square rule. And this talks through how light spreads out over an area as you move it away. So it's not directly proportional. This follows the inverse square rule. Worth going away and just looking up and reading a couple of little notes on that. And then temperature. We could just increase the room temperature. Artificially increase or decrease, couldn't we? And see what the impact of that is. Okay, so we've got the movement of water, and now we need to talk about our final little bit. So we've talked about xylem, we've talked about what's happening in the xylem. So final page. I'll actually find a page. Final page from me. We're now going to talk about what's happening in those phloem. You're doing fantastically well. 9 minutes left. So we'll just very quickly go through what's happening in the phloem, and that is the process of translocation. So when we do photosynthesis, we're going to have some sugars produced, and those sugars are mainly sucrose. And I guess for reference, if you take a glucose and add it to a fructose, you create sucrose. And this, along with amino acids, are transported around the plant. And these two together are generally referred to as cell sap. And the transport of this cell sap is in your phloem. And these are slightly different because they are living cells. So remember, our xylem were dead, the phloem are living cells. And they're transporting in different directions. So whereas the xylem is very directional, the phloem are transporting to where they are required. So we're transporting dissolved sugars to be used or stored. Maybe they're going to be used for respiration, or maybe we're going to store them. Sorry, James, 1 second. Mr. Stevenson's just come to check up on you. Be back in a second. Right, so it says, well done. Thank you for being here for so long, gents. Right, we're nearly there. So we've got these phloem, and they have a slightly unusual structure. So let's just have a quick go at drawing that structure. So often see it represented. We've got our tubes, and then in between them, we have this structure here, and then I've got another tube running alongside. Again, I've got this slightly unusual structure here. And in here, I've actually got some organelles. And that is because this bit here is called a companion cell. We've then got these little structures here that are called sieve plates. Then got the cytoplasm. And there are gaps in this cell wall of the sieve plate, which are going to allow things to pass through. This is one cell stacked on top of another cell. We've got this sieve plate with some gaps in, and that is what is going to allow our sucrose and amino acids. And maybe on this side, they're all traveling up this way. But on this one, they're going in a couple of directions, heading to some cells here, heading to some cells here. And so you've got movement in different directions. So if we finish off then with a final little comparison table. We compare our xylem and phloem. It's quite a common question. Similarities, differences, or in this case, differences. We've got the type of tissue. We've got what is being moved. Got our process. So what is the process called? Got the direction of flow. And then we've got any important reference of the cells. So xylem and phloem. So have a quick minute now, see if you can just fill that in. What is moved? Well, in the xylem, it's water and mineral ions, and in the phloem, it's sucrose and amino acids. Process name is transpiration, and actually it can be referred to as the transpiration stream because it's a constant stream through the plant, but the movement of sugars and amino acids is translocation. Direction, one way, and all directions. And then finally, this is dead cells, and this is living cells. There's a bit of a whistle-stop tour. As I say, there's definitely some things you want to go away and read up on, maybe look at a bit more detail on the photometer and the inverse square rule. Have a little look at that practical, maybe see if we can see some data online or some data questions. What I'll do is, as a thank you for joining today, I'll print you some exam questions. I'll give you the questions, and what I'll do is I'll send home the answers to your parents. So I'll say a little thank you for joining, and then I'll send them the answer pack, and so you can say to them, okay, I've done all the questions. Can I go through the answers now? Absolutely fantastic. Well done for joining, gents. I hope you enjoy the rest of your evening. Take a little break now, and then maybe in half an hour to an hour's time, have a little go at rewriting these notes, see what's gone in, see what hasn't gone in, and then try rewriting and answer some questions again. But I'll make sure I get those questions to you in assembly tomorrow. Thanks very much for joining, and same time next week, please. Really well done. Thank you.